Any questions for me about the essay assignment exam? So only A's, no B's, no C's. Okay, good. Let's see. So I've got a few not drafts, but few plans from students that asked me to check them out. It seems like you guys understand what the hell is happening, so that's good. I'm thinking that probably everyone has the same idea, right? You went through the CRISPR screen and then the 2 genes of interest. Some of you went to a third one and things like this, right? So if you're not doing that, then maybe review. What else? Yep.
When it comes to the methods section,
Yeah.
there's obviously a lot of stuff that's actually in the methods of the manuscript that isn't super relevant.
Exactly.
I assume you just want us to be talking about what actually applies to, I guess, the experimental design of what we're talking about in our results?
Exact. You know, I don't want you to get so specific that you tell me about the buffers. But at the same time, the CRISPR screen, for example, is important, right? And so probably no one in the class, or maybe a few people in the class knew what CRISPR is. So the general level, at this level, you don't know what CRISPR is, right? So you have to explain it a little bit more in detail, because you have to imagine as if you were writing to someone, to yourself prior to taking this class, okay? And so you can use references from other papers. Probably you can even find the paper where that technique was actually developed and use that paper. And if you look in the paper, I'm pretty sure they refer to it somewhere. And so you can use that. Obviously, if you're looking at depth map and things like this, what the hell is that, right? So, you know, define those, explain them a little bit. The results, just describe them, the statistical methods, describe them, and then in the discussion, don't get too vague in the sense that don't overdo it for the clinical potential, for example, of these findings. It's the purpose of this assignment is to tell me what they did in their work. Obviously, it's going to be kind of a boring end if you don't expand on it, but don't go like a full page just on the clinical applications or whatever future research this can give, okay? Anybody following the Nobel Prize stuff? No? So there's a lot of, you know, the first day everyone was happy, but now there's a lot of scientists that are mad because non-chemists are winning the chemistry Nobel Prize. AI people are winning the physics prize. So it's getting a little bit mixed up in there. Today we'll see why AlphaFold is important and why they won the Nobel Prize. But I think there were other people that could have won the Nobel Prize other than these guys. Not because it's just a weird way to win the Nobel Prize if you're a CEO of DeepMind and then you end up with a chemistry Nobel Prize, but there are a lot of discoveries that have a bigger impact than AlphaFold itself, right? In any case, so before I get started, I was supposed to answer a question from Alex, which asked me, is the 90% accuracy for Oxford Nanopore per base or per sequence? And it's actually per base. So the companies that do the sequencing, like Illumina and all that, they base their accuracy using the per base accuracy method. This way you can compare them between each one of them. Although one thing that was a little bit different, it's better to have a 90% accuracy per whole fragment than to have a 90% accuracy per nucleotide, because what happens is that you add up those and then it ends up being a much smaller percentage in accuracy. Oxford Nanopore and PacBio are not that precise. Nanopore, because the electricity reads, so the current reads, the current first is so small, that's the first thing. And second, usually it's read into 2 bases, and sometimes there's interference in there. That's why there's this issue. Also, so for these methods, because the read is done directly on the sequence, sometimes also the sequence moves too fast into the pore. And so one of the major things that they're working on is improving their motor protein to slow down the flow of DNA through the pore. The thing is, each of these technologies have a an advantage and disadvantage, right? And so you have to use all of them together to kind of get the closest, the perfect sequence, let's say. Because if you're doing, for example, repeats, you have a lot of repeats in your sequence, you're not going to do short read sequencing like Illumina. Instead, you want to do something like PacBio or Oxford Nanopore, because they allow you to read a longer sequence, and so you can cover the full repeat region plus left and right to it, the adjacent regions, and then you can use those to map. While if you're doing Illumina, you could just basically cut inside a repeat and you'll never know what the hell is coming here and here, right? And that repeat could be mapped to another region, so you don't want to do that, okay? I'll also review a little bit what I talked about last lecture. So the lectures are kind of similar. So last lecture, I was, at the end of the lecture, it started being a little bit weird because didn't get time to review properly, but so I was a little bit everywhere. So I kind of covered a little bit of today's lecture, but I'll still redo just a small review just so that you have everything in place, okay? So prior to last lecture, we were able to sequence the human genome. We learned how to sequence it, the different methods we used. So then the idea after sequencing the genome is to actually understand what the genome is made up of, because its complexity has increased. And so this complexity increase requires, further investigation to see why, for example, in yeast we have less introns in genes compared to humans, and why do we have this sequence of DNA that we don't know between genes and things like this, what it does. And so the first starting point is usually always the simplest one, and this is usually the proteins, so genes that produce proteins, because those can give you something that you can work with. While if you're looking, for example, at enhancers, silencers, things like this, it's quite complicated to study them because changing the way they are structured will affect function, but you will not be able to link it to something, right? It will be very hard for you to link it to something. So genes that code for functional proteins are usually the starting point to understand any genome. And since we have some DNA sequence from other organisms, then we can do some comparative genomics to understand what is similar between these organisms and what is different, okay? The enzymes in glycolysis are pretty much the same between organisms, so if you want to find all the 10 enzymes in the 10 steps of glycolysis, then you take them from yeast and try to figure out if there's sequence similarity between yeast and humans. And you'll see that there is sequence similarity, for example, hexokinase. Then you'll see that, for example, yeast has much less introns in its sequence. And so when you compare it to the human version of hexokinase, you'll see that the sequence from yeast will overlap, but there will be spaces between. So you can then test if those are introns and things like this, okay? So when we talk about proteins, which are the basis of our first analysis in discovering what the human genome is made up of, then there are these structural modules that you can look at. And this you probably covered in many other classes, but in genomics we're interested a lot into domains and motifs. And the definition for these changes depending on what field you're working in. Functional genomics will not look at a domain as a structural piece, but it will look at it as a functional piece. And so this here changes the way in which you look at it. For us, genes contain, so for everyone, genes contain domains and motifs. For example, you can have DNA binding domains. And these DNA binding domains, all they do is that they take that protein and they anchor it on a specific sequence of DNA, okay? A good example is transcription factors. Transcription factors are proteins that are found in the nucleus, and they direct a transcriptional program based on where they bind on DNA. For example, let's see, what is a simple one? The androgen receptor. The androgen receptor has a binding site on DNA, and every site that is targeted by the androgen receptor. So it's called receptor, but in reality, it's also a transcription factor. It just receives the androgen. And so it binds to many genes. I don't remember how many, but thousands of genes that direct the androgen program, okay, the androgen transcription program. And this is important in many diseases. For example, if we're looking at prostate cancer, then we can talk about the androgen receptor, how it is involved in there. Because prostate cancer has two major types, the androgen-sensitive prostate cancer, which reacts to androgen therapy, and then the androgen insensitive. And as you can see, because it is insensitive to the androgen, then the transcriptional program of the cell will change because the binding of that transcription factor will change which genes it binds to, okay? So this is an example of transcription factors that have a zinc finger, okay? There are other, obviously, types of domains that bind to DNA. Now, domains happen in multiple proteins. For example, if you take a look at kinases, kinases have similar domains and they have similar motifs, and their goal is to add a phosphate group to something, okay? One interesting thing, though, is that some domains do not always have the same function depending on where they're found in the cell or which organism. So a zinc finger domain could be involved into binding DNA, let's say, for humans, but for yeast, it binds something else, okay? So it's not necessarily functional. An example of this, just from an abstract sequence, if you remember last class, we talked about the blasting tool. So BLAST-P allows you to align or to kind of search for sequences that are similar to yours in all the database of known sequences. And you can find, for example, let's say these are different proteins from different organisms. You can see, for example, that there are conserved domains here. There are conserved amino acids here, and then the rest is a bit different, okay? So from this, you can see what is the consensus sequence because all the organisms share this part here. And so then from there, you can build a pattern. This pattern here that is built is basically telling you there's a K, RK. I forgot what the K is. R is arginine. And then these two here could be any amino acid, and then continues like that, right? And so this pattern here, then when you're looking at other organisms and you find this specific pattern, you can somewhat confidently say that this is X domain, okay? Zinc fingers have their own pattern, kinase domains have their own pattern, and so you can use these tools which have the sequence of proteins in them, take the sequence that you got from, let's say, a gene that you don't know, and then look for these patterns. Obviously, you don't do them by hand. Before, we used to do this by hand, but now you use computers. And it will identify each of the domains of that protein and tell you what their potential structural role is, but also functional role. Here's an example from a protein, from an amino acid sequence that was basically produced from research. And then you can use two different tools to see where the domains are. In this case, your user sequence that has 422 amino acids that you don't know what it does, you can put it into the tool, just run the tool, and the tool will give you where the domains are. For example, this homeo box here, and there's pair 2 domain. Obviously, you'll get the description of the homeo box, what it does, and the paired domain, what it does. But this does not mean that they have the same function between as what they're saying here. This is predicted. Now what you need to do is actually test this out. So you need to remove this part here and see what actually happens in the cells. And you can see with another tool, some of them just will give you, they'll both usually give you the domain itself, the sequence of the domain itself, and the score of similarity to other domains that were found in other organisms or other genes or whatever. If you look for a kinase domain, you'll see that that kinase domain is very similar, and it will tell you a higher similarity score. What else? So that's that. When you're looking for domains, there's a lot of tools. It's like there's like tens that come every day. Okay, people just keep creating these, and there's so many of them that you can use. Usually, you build this tool using some sort of bioinformatics, and then the bioinformatics tool, the data. So not the data. The program will be published online, and then you'll have a paper that comes out to describe what this tool does. And then the team working on it will release a new paper every time to give updates about this tool, okay? If you're looking for function and location of the protein, then you can use these tools, for example, and you will put your sequence, and it will tell you what the domains are, the functional domains are. For example, this is an output from SmartScan, and this STYKC domain is a phosphotransferase. So if you run this through the database, let's say, for example, you sequenced your human genome, you find a gene based on some parameters like the ATG start site, the stop codon, maybe the TATA box in the promoter, then you know that this is potentially a gene. Then you want to find what domains are found in that gene. From there, you can infer function, because if you have a phosphotransferase domain, probably it's because this protein has a function to transfer a phosphate from one thing to another, right? Obviously, this is still primitive information because it's all predicted. You still need to do more research on this using some experimental methods, okay? You can try them all. Just write on Google bioinformatic tool to find domains, and you'll see a lot of them, okay? Right. For us, structural genomics people, we're interested in domains in terms of structure. So we'll actually be looking at pieces of DNA that fold, that produce proteins that fold, so pieces of proteins, that fold into a specific pattern independent of the whole protein. So if you were to cut that piece and put it somewhere else, it will still fold in the same way, okay? This is what a domain means for structural biologists. An example, again, I keep repeating the DNA binding domain. This is a very important domain, and there's many of them. And if you take a look, for example, here, just to give you an example. So this is AlphaFold. This is the technology there that won the Nobel Prize. So this is a protein that I'm working on right now. And if you want to find, for example, the DNA binding domain, you can first go on UniProt. And I'm just giving you examples of how you can do this, and then you can do your own research to see what's happening. So if you go to UniProt, you go down until you see, let me just search it because we don't have. So, wait, where are the domains? 1 second. Oh, there you go. So if you want to look for the DNA binding domain, so this is the zinc finger that binds to DNA, then you have to find the sequence here, right? And zinc fingers have different numbers of cysteine residues. For example, this specific one is of the type that has 4 cysteines. Just an example of what's happening in real life. There's one here, one here, one here, and one here, and these are important for binding the DNA, okay? These cysteines will bind to a zinc metal, and then they will allow for binding to DNA. So if you want to find the structure of this motif here, then what you could do is go to AlphaFold, put the protein that you want, and then find 79, right? So this here is your domain, okay? This is your DNA binding domain, okay? What does this tell you? Not much if you look at it from here, but now you can also, soon you will be able to put a sequence and modify one of the cysteines and then see how the protein changes. And that change, you can then study its binding to other proteins and things like this, okay? For example, in my work right now, I'm studying its ability to bind to DNA based on hydrogen peroxide levels. And so if you put hydrogen peroxide, you will basically break those cysteines, and then the DNA binding ability of this protein will change. And because it changes, it will lead to different pathologies, okay? Why hydrogen peroxide? Because in cells, for example, if you have diabetes, you'll end up accumulating oxidative stress, and oxidative stress will lead to further problems, and this is one of the major players in controlling that. And so obviously you have to study this protein in that sense and then try to alleviate that problem to see how you can fix that. Okay, that was my initial project. Now I moved a little bit to cancer, so not dealing with this anymore. What else? Domains happen in proteins. So domains don't just pop up randomly. Sometimes there are duplications that happen, and then those duplications will lead to moving between genes. So you'll end up having, let's say, for example, one exon going from one gene to another, and then suddenly that new gene, that second gene, has a kinase function, but it did not have it before. So this is just due to evolution and the way that crossovers happen in cells. Okay, so that's that. What's the difference between motifs? By the way, if you blast, this will not be sufficient to give you the protein domain. It will tell you that this sequence is similar between them, so potentially that's the domain, but you'll realize that when you actually take the 3D structure, sometimes they don't become the same structure because of the whole area in which that sequence is found. Okay, and so BLAST does not give you the full domain. It just tells you that these pieces are similar, so potentially they are domains. Motifs. So you'll see people always interchangeably using motif and domain. Motifs are usually smaller than domains. Not usually. They're always smaller than domains, and they are part of the domains. Okay, and these motifs have, it's like the smallest basically sequence that does a certain function. So motifs play a specific role within the protein, within the domain. For example, this cysteine, aliphatic amino acid, and then X means a certain amino acid that comes right after, is important for modifying cysteines to add lipids to them, so prenylation. Okay, so motifs usually are incorporated within domains, so you'll have a domain that contains multiple motifs, and then domains are then found into proteins, so you can have multiple domains in a protein. Okay, phosphorylation site of a protein is a motif. So phosphorylation happens at the serine, threonine, tyrosine. So if you find a serine, threonine, tyrosine, potentially that's a site for phosphorylation. Okay, so this is what I mean. Let me give you an example of this. There's a protein called PTEN. PTEN is a phosphatase, right? And it has a cysteine inside its catalytic site. So if that cysteine gets oxidized, for example, so the cysteine is a motif. If it gets oxidized, then the protein does not have the phosphatase function anymore because it changes the, closes basically the catalytic site, right? So that's the motif. The domain is the full catalytic site. The motif is that little cysteine in it, and that cysteine, when it gets oxidized, changes the domain, closes it, and that leads to its inability to have a phosphatase activity. Now, PTEN, just an example for PTEN. PTEN is the cause of many cancers, so you can search about this. It's also important for glucose metabolism, so it's involved in insulin sensitivity and things like this. So this is a very important protein, and just a small change in one cysteine in the catalytic site does a lot of problems. Okay, so if you find, for example, a protein that you blast, find another protein that looks similar to it, and there's a region in it that is similar between both proteins. For example, you find that that region is similar to PTEN. Then you look at it and see, is it part of the catalytic site, for example, of PTEN? If you do find that, then you know that potentially your new protein that you found has a catalytic site that has a phosphatase activity because it's similar to the sequence from PTEN, right? And then you look for a cysteine in it because you know that PTEN has a cysteine inside that catalytic site. If you do, then you might think that that motif is the one that affects how open and closed the catalytic site is. And then you can assign a function to that protein, which is being a phosphatase. Predicted. This is all computational. We didn't talk about experimental yet. Experimental, you would have to, for example, change that cysteine to another amino acid and see what happens with the catalytic site. Then you would, for example, try to oxidize that cysteine and see what happens with that catalytic site, right? And you can do this in a tube. all you have to put is some proteins that are phosphorylated, and then see if the phosphatase activity happens there or not with your protein that you, for example, can attach on a bead. Okay? Make sense? Simple. Okay. Instead of using proteins or domains, you can also take a look at the sequence of transcripts that define genes. Because, yes, yeast has a lot of the function that is found in humans, but not all of it. So sometimes it's hard to find genes just based on what we know from yeast, knowing that humans are much more complex, right? And so one way to work with this, to work around this, is to use transcripts. So if you look at the central dogma, you have DNA, and then RNA, and then proteins. So RNA are pretty much, mRNA is pretty much similar everywhere, in the sense, not between organisms, but I mean for proteins. And so you have always that poly A tail that is found at the end. So you could technically look for regions that have poly A tail, and then you would identify that those transcripts lead to a protein, right? But sometimes they don't. So this is relevant for example, pseudogenes, which I told you about last class. So pseudogenes are sequences that look like genes. Sometimes they produce the transcript, but not the protein. Sometimes they don't even produce the transcript, okay? And so currently we've been talking about DNA sequencing technology, but there's also RNA sequencing technology. So you can just do an RNA-seq experiment, and you will find all the transcripts that have the poly A tail. When you have the poly A tail, then you can then map them back to your DNA and then figure out if these produce proteins or not. Okay? So let's say we found all the genes using these computational methods. Then obviously we have to find their function. So I gave you just a little bit of examples, the past few minutes. But there's a lot of there's a lot of information just in the sequence itself, right? For example, the functional domains can be helpful. I just talked about the kinase phosphatase domains. And then you can have different types of domains, general or specific. Like right now, the P10 that I told you about, that's quite specific. But you have other domains that are more general. And these are the examples, okay? So you have specific domains, for example, a kinase domain. So if you find that there is a kinase domain in your protein, then you can directly put it in the group of kinases, right? If you find a protease domain, a domain where the site in this protein will break other proteins, for example, trypsin will break different proteins into smaller pieces, so then you can put it into the proteases group, phosphatase domain, like the P10 protein, P for phosphatase in P10. And then you have also the more general sequences based on your alignments and all that. For example, the transmembrane domains, which allow a protein to be incorporated into the lipid bilayer. Protein interaction domains, they're not specific because what protein does it interact with? So there's many different ones. DNA binding domains, same thing. They're less, although DNA binding domains are starting to be found to be more specific because each DNA binding domain will bind to a specific motif, okay? Let's say you want, as a scientist, to, so let's say, for example, the government puts this project on their website, so you can find these online. I don't have the website, I forgot what the website is, but they will ask for people to submit plans of how to solve a problem they have. For example, measuring the levels of an acid in water, okay? You, as a genomics person, you can start playing with proteins to achieve this task, right? So one way to do it is to just use chemistry, but there are better ways that are more clean, more environmental friendly, for example, using biological sources, right? And so you can take, for example, a bacterium and then find a protein that binds to that acid or that reacts with that acid, combine that domain, that binding domain, to a transmembrane domain. So now you have that acid binding domain with the transmembrane domain, and then inside here you can have, for example, a kinase activity, right? And or a phosphatase activity, right? And so whenever there is a binding of that acid, then there's a change in this structure here that's transmembrane that activates, for example, this phosphatase. And phosphatase breaking down phosphate groups everywhere in any protein will release these pyrophosphates, right? This is something that we can measure. So this is an example of where you can use these domains in real life and actually build your new proteins that you want to do, and then you can apply it to measure things, whatever is required, okay? I'm giving you this specific example because there was a project that was put online by the Canadian government to measure this domoic acid, and we had applied, me and a friend, to do it. We almost got it, so it's based on something even simpler than this. Still working on it, well, not me, but the other guy. So I can't get into it, but it's based on cells engineering proteins that don't exist in, that do exist in other organisms, but making a hybrid protein, let's say, and then measuring some sort of change, electrical change, let's say, in the in your tubes. Okay. So this is pretty cool stuff. As you can see, I love synthetic biology. There's a new center that opened here in the department, not in the department, but in the new university, that did genomics, the genome foundry and synthetic biology. Forgot what, CSFG or whatever, okay? Just look it up, okay? So they're doing pretty cool research. If you're thinking about your future and you are not interested in cancer like everyone, that's a pretty good spot, because then you can, build your own startups much more easily compared to just working on something that everyone is working on, and actually maybe even do something crazy, okay? So think about that. What else? There are also domains. So I'm talking about all the types of domains, not all, but most of them that exist. So location defining sequences. So if you, for example, do your search and compare the domain that you find in your unknown sequence to something else, and you know that that's a hydrophobic region, you can infer from there that probably it is a transmembrane domain. There are also domains that are that encode a signal peptide. So the signal peptide can tell you that probably this is a protein that is secreted, or also that goes to the membrane. NLS, this one, it brings the protein to the nucleus. Could be potentially a transcription factor or some other type of protein. Peroxisome localization signal. And this one here we just talked about, the motif that leads to prenylation. So this could tell you that maybe it's important for membrane attachment, because if you have this motif, then you're adding lipids, so could lead to membrane attachment of your protein. Okay. The types of assays that you can do to find function, there's millions, I think, every variation of possible assay that you can have. So in this case, we're talking about experimental work, not structural bioinformatic work. And so you will use different types of assays. If you're talking about a kinase, then you'll probably be thinking about enzyme activity assay, some sort of luciferase activity or something like this. If you're talking about a zinc finger domain, you'll probably be studying metal binding in your test tube. If you're looking for its expression, then you'll be looking at transcriptomics, proteomics. Localization, you'll be looking at its expression into different tissues. So you'll do probably histology here. If it's cellular localization, you'll probably be doing, again, histology, because you're comparing the different cells in different tissues. And then if you're looking at intracellular, then you'll be using microscopy, right? So if your protein, you see it, so let's say, for example, you go to a microscope and you're trying to see if your protein binds to DNA, then you'll stain that protein with some sort of tag that turns it green. And then you'll also tag your DNA using some sort of tag like DAPI, whatever, which is a stain for DNA. If you see a lot of dots of that protein on your DNA, that probably means that your protein actually acts on the DNA, right? And it also probably means that it has an NLS because it localizes in the nucleus. And so if you do your structural studies in terms of sequence, then you'll probably find an NLS sequence as well as a DNA binding motif. Okay, so what else? Then you can also do mutant phenotypes. Actually, this is important in any study that you're doing. If you want to talk about the function, you need to break the function. So if you don't break the function, you cannot say that this is the function of that thing. And then you need to rescue it. So usually when you're doing research, you do a lot of rescue experiments. You break a protein and then rescue it in some sort of way so that you see if your wild type, if it goes back to the wild type setting. You can take a look at the interactions of that protein between them, usually through protein binding domains. Proteins, as you know, form these dimers, tetramers, and things like this. They also interact with other proteins, so you can do this kind of work. Anybody in the lab doing any of this in research? Specific lab? No? Jeez, everyone's sleeping.
No, we just don't do research.
No? Yeah, the program is quite theoretical. That is true. So if you're doing a master's later, try to go more practical. You have all the theoretical there, but get into the practical. There was a student that asked me about, well, not asked me, but we were talking about the future perspectives of biology. As a professor, I have to say it's amazing. But in reality, I think I'm going to do a little, like 30 minutes or 20 minutes at the end of the semester to guide people. If you're aiming to become a professor, review your aspirations. Not because you're not good or you're amazing or I don't know what. It's just because I'm in it, and so I realized that actually it's quite saturated. So if you want to teach here in Montreal, it's going to be very hard as a full-time, not talking part-time. And I should have known this before because I see the department and there's like people that have 12, 13, 14 years as part-time. Like, how arrogant can you be in thinking that maybe you're going to become professor before them, right? So this was kind of the thinking, in not really like, oh, I'm going to be, but not them. It's just I never thought about this, right? So if you're thinking about teaching, be sure that you're able to be mobile. That means move and change country, because people usually what they do is that they will study up to the postdoc and then they start looking for places, and whenever they find a place, that's where they go, okay? So that leaves you with PhD, masters, or switch fields. So if you're looking to work into biotech sector, I suggest doing a master's and then going to get some experience, then maybe the PhD, okay? Because there's people, the PhD does not add a lot in terms of biotech sector, other than it makes you kind of lead projects, but before that you can also just go the experience route, okay? And if you're doing a master's, try to get more practical than theoretical. This is one of the mistakes I've done. It's there's people that do research just to do research. Well, not really done, because if you're doing research on something that will have an effect in maybe 200 years, then right, what's the point? But if you're doing research on something that is less theoretical and has a direct impact, there's a higher need for that in the sector compared to actually theoretical stuff. Because there's profs that do 40 years of research on the same topic, they keep publishing, they get the money, it's amazing, life is good, but it has no impact. And so because it has no impact, I myself don't see a point into it, and society does not reward things that have no impact, okay? If you see society, if you want to be rewarded, you need to bring value. Doctors bring value, so they are rewarded. But if you're a theoretical physicist, almost nobody cares, right? And so this is what I meant. If you're trying to do research, try the synthetic bio, even though at the beginning it might be a little bit weird, but it's good for you. At the end of the semester, I'll also talk about, I'll talk about all the scholarships that you can take and things like this. Because I know that, I get questions from students, but also I know that I was pretty lost with this stuff until I figured it out. And in our field, you don't know things, right? You have to figure things by yourself. It's like you don't learn how to become a professor. You just become a professor, you know? You did a little bit of TA, a little bit of pedagogical training, like 4 lectures, and that's it. You're officially a prof. But in reality, it takes more than that. And for the scholarships, if you don't know when to apply and you start a master's, if your PI doesn't tell you about it, then you end up missing that deadline. And because you miss that deadline, then you become less competitive and things like this, okay? So I'll do this, talked about this last time with a student. So I'll do this maybe at the end of the semester so you guys have an idea. But yeah, back to biology, back to this. So there's a problem though. When you find your proteins, which are encoded by genes, sometimes you'll find multiple copies. And these multiple copies might make you think that they have the same function because it's a copy of the same gene, right? But they don't have 100% similarity, and those changes will lead to actually fully, completely different functions. This mixed me up a lot in my studies: homolog, ortholog, paralog. Homolog is the main category. It's proteins or DNA sequences that have similarities between their sequences, and then there's 2 types. Orthologs happen between different organisms, so mice and human. If you find a gene in mice and human that is similar, those are called orthologs. Paralogs is within the same organism. It's a duplicate of that gene, right? For example, the globin gene had a duplication 500 million years ago, and it led to myoglobin and cytoglobin, okay? This is called a paralog because they have similarity and they're within the same organism. And these paralogs then went through speciation between mice and humans about 100 million years ago. And so if we compare the myoglobin between humans and mice, we can call them orthologs. Myoglobin and cytoglobin of human, we can call them paralogs, okay? So when you do your BLAST or whatever, and you find the same sequence but in the same organism, sometimes you'll be dealing with paralogs, so duplicates. And gene duplication seems to be kind of weird, but it happens a lot, okay? This is a feature of evolution, and it happened actually many times, okay? So I have a question for you. Which one gives more information about the function of a gene, orthologs or paralogs? Sorry? Orthologs, why?
Because it has conserved functions that is essential for the organisms.
Yeah.
Paralogs are caused by mutation, they're not driven by.
Yeah, so good. Let me just write. Sorry guys. Yeah, so yes. Exactly. So globin, oxygen binding. So myoglobin, it's important for the oxygen reservoir in the cells, so it kind of stores oxygen. Cytoglobin is not as well known as myoglobin, but its function is to protect us from oxidative stress. So it does interact with oxygen, and it also can serve as a reservoir of oxygen. So what the 2 students said, the gene of interest to infer its function, you usually look at orthologs, because if you have an ortholog in another species, that means they probably do the same function in both organisms. And if you compare their sequence, you'll see that they're more similar compared to paralogs. Paralogs usually have less similarity compared to orthologs. And that just means that they kept their function through speciation, okay? What else? Paralogs usually appear because you have a duplication event, and after that duplication event, the new gene, the new copy actually takes on a new function, or its function changes a little bit, okay? So obviously you guys looked at the next slide. I should remove these. Let's take an example here, okay? Let's take a look at the globin genes in human, zebrafish, and mice. if you're comparing these orthologs here, you can see that their sequence is quite similar. They have some differences in here, but mostly similar. From here, you can find all the domains, right, of the hemoglobin. And then if you're looking at paralogs, so paralogs between this inside the same organism, so let's say, for example, the hemoglobin of human, hemoglobin A and B with human cytoglobin, you can see that there's basically almost no similarities. Okay. What else? How did this happen? So in the evolution up to humans, so the vertebrate evolution, we had an ancestral globin before. There was about 800 million years ago, there was a split to neuroglobin and cellular globin. If you think about neuroglobin, you can start maybe imagining that this split here happened when the first primitive nervous system appeared. Then you have your cellular globin, which was then split into multiple different globins. So this happened when there was a split between the jawless fish and jawed fish. Just to give you an example of evolutionary biology, that gave to hemoglobin and to the ancestor of myoglobin and cytoglobin, and then about 400 million years ago, or maybe 5, there was another split for cytoglobin and myoglobin. So these don't happen just like that. Usually there is a genome duplication, segmental duplication, or gene duplication, right? And that's basically this slide here. So whole genome duplication seems a bit weird, but it's just the duplication of the full genome. And this usually happens due to, for example, problems in DNA replication and segregation, right? But it's still not understood very well. But whole genome duplication will lead to an increase in complexity because some genes in the whole genome duplication will change function. Some paralogs will be lost. Others will remain. And so this is due just to the fact that the paralog that was added does not add anything. If it has a new function due to mutation that makes the organism more fit, then it will keep that paralog. But if it does not have any mutation in the new paralog that makes the organism more fit, then that paralog will be lost. We had many whole genome duplications that happened through the evolution of vertebrates. So lancelet is the Amphioxus, Younes the tunicates. And so there were 3 of them actually that happened before us. These are 3 major whole genome duplications that happened. This is why some genes that we have in multiple copies, others not. For example, one of the protein that I'm working on called Ronin. This is TAP11, so you can imagine that there's TAP10, 9, 8, right? And so there were many copies of these genes. Every time there's a duplication, they get multiplied, whatever you have in there, and obviously they all kept their functions because they had new functions, each one of them. Okay, Ronin is important for development, embryonic development, and cancer also, because if you talk about development, cancer gets involved. There's also segmental duplication. Segmental duplication is a bit different compared to whole genome duplication. Segmental duplication, only a segment of the genome gets duplicated, and so when you're talking about the assembly during your sequencing, there's something interesting that happens. For example, if you have a unique segment or a duplicated segment, it will affect the quality of your reads for sequencing. So unique segments for an autosome will lead to 47 pieces of DNA that were sequenced that can be mapped to any given 5 kb fragment. So if you take 5 kb fragment, you'll usually cover this piece here, or parts of it 47 times, okay? This is just because you have multiple copies of the genome, and so when you're sequencing the multiple copies of the genome, some sequences are similar and some are not, and so on average, for any 5 kb fragment, you'll have 47 pieces of DNA that will map to it in your sequencing effort. If you have a duplicated segment, this increases to about 5 times. 5 times, that means that because you have a lot of duplicate sequence in your basically your gene is found twice in your genome, this will increase the number of reads that happen that are mapped to that 5 kb fragment. For the X chromosome, for example, X chromosome is one of the most, let's say, less repetitive chromosome of the human genome, and so you have less reads that can be mapped to the 5 kb fragment. If you look at the Y chromosome, this is probably much higher because it's full of repetitive sequence. Okay? Examples of recent segmental duplications in human genome. So these are kind of these shapes here, just link one part of a chromosome to the other, telling you that basically it's the same sequence. This is due to segmental duplication. You can see that chromosome 1, which is the biggest, is one that has the most segmental duplication. Chromosome 7 is similar too. And so these just tell you which sequences happen twice in that chromosome due to segmental duplication. You can see that chromosome 7 has a lot of that. Okay? So you can imagine that this one was very hard to sequence. If you look at the Y chromosome, it's like everywhere. Okay? So that's that. Gene duplication. Sometimes also just the gene gets duplicated. And this happens due to many things, but including crossovers and things like this during DNA synthesis, replication, and then separation between 2 cells. And so this is a very important factor in organism of evolution, because gene duplications lead to adaptations, because the new gene will be selected, will gain mutations that will lead to improved or different functionality. This leads to also the creation of gene families, because you have these multiple copies of the same gene that are a little bit different. For example, nuclear receptors, which are transcription factors. This is a huge family of transcription factors, and they all have pretty much similar parts. Usually what changes is their DNA binding domain, because it binds to a different segment of DNA, and their protein binding domain, where it binds to other factors. Okay? But the main structure remains the same, probably resulting from gene duplication, segmental duplication, or whole genome duplication. Gene duplication creates pseudogenes, because what happens is that you have a duplication of that gene, but then wherever it gets inserted, that duplicate can lead to something that looks like a gene, but actually does not produce any transcript. And this is just based on the sequence that is created from the duplication. So it's not a full duplication, let's say. And what else? Most of the time, duplicated genes will appear in clusters. These are called arrays on the DNA. So they will be in the same region, but then some of them will be dispersed. For example, the ribosomal genes, the ribosomal protein genes, they're not almost similar, but there's like 100 of them, and they all have little variation in the function, but in terms of protein itself, they're quite similar. And this is a type of protein that is dispersed throughout the chromosome. So why am I talking about this here? It's because we're trying to understand the structure of the human genome, right? And so sometimes you will be finding many copies of the same gene. And so to understand their function, you cannot just assume that they have the same function. They could be paralogs, and because they're paralogs, you'll have to use all those experimental methods to understand what's the difference between them to get their function. Many tools. So I talked a lot about this. You probably, if you're taking functional genomics, you're probably tired now of the number of tools that you're learning. There's a lot of them. So just there are tools that require machines. Already there are a lot of them. But in terms of tools that just require the computers, it's hundreds, thousands, okay? So you'll find so many tools that each one has a different way of doing it. And even if you analyze the same sequence using 2 different tools that say they do the same thing, they will give you different results. So this has created a lot of variability and a lot of inconsistency in research. And this is actually one major problem, because we keep producing this data, but most of it is actually not reproducible, right? And it is not reproducible because of the poor design of these softwares. That's the first thing. And also because of the lack of validation. So I'll give you an example. Let's say, for example, you're doing research and you're studying a protein, and then you run that protein sequence against a database that tells you what its function is. In 2010, the function could be this, but as we gain more data, if you rerun that same sequence, you suddenly have a change in its function, right? And this is a good example for if you take a look at transcriptomics, where you're studying the set of transcripts in your cell, extract RNA, sequence the RNA, and then run it through an annotation software. It will tell you, for example, that treatment X led to a change in the RNA set in the cell, which is, for example, a sign of cancer because all these transcripts are now upregulated. Now, if after a few years you come back and you run the same data using the new database, the more updated database, that function can actually change, right? And so a lot of papers that were published previously become obsolete because of this kind of thing. And the speed at which this is happening, it's like after 1 year, your data does not make sense anymore, right? So if you're Googling this and you're using these softwares, be careful with it. Try to validate between each one of the softwares. But there's a lot of them, okay? You have softwares that study transcripts, regulatory regions, how open or closed the chromatin is, RNA-seq, ChIP-seq, whatever seq you want, okay? Some more interesting tools that make sense are comparative genomics tools. These allow you to compare your sequence with other organisms. So usually this is backwards. So because it is backwards, you end up finding something that makes sense because that was already studied in the past. And so you already have an idea where to go. But if you're going forward genomics, so you're going from a smaller organism trying to find the equivalent in a human genome, for example, then that might find you something, but it doesn't mean that that's what it does, okay? And as you might know right now, you can use CRISPR to break everything in the cell and then find the function. This is a Nobel-winning prize that is going to pretty much change how we do medicine, okay? It will be used for so much, it will be used for a lot of the gene therapy approaches to treat different diseases. For example, if you're studying, I don't know, a CFTR gene, which is the cystic fibrosis gene, you know that just removing that gene and replacing it with the right copy using CRISPR, which is very simple to do, right, will cure that person. So, well, not cure that person, will put the proper copy of the gene in there. Now, how do you cure that person? That's another story. But CRISPR is used to study a lot the function of proteins, because when you sequence, for example, a gene, and you want to see if a piece of DNA has a certain function, then you can remove it using CRISPR. And then if you're looking, for example, at a specific motif, let's say in the P10 protein, you can change that cysteine to something else using CRISPR. So it can just change one piece of the catalytic site. So this is a crazy technique that just keeps getting better and better. And you saw a little bit of this in your paper, the prostate cancer paper. So that's that. What else? This is just a graph or figure that shows you what I mean by the divergence of duplicated genes. So usually just because you have a change in one amino acid could actually change the function of that protein. I just showed you an example of P10, right, where you lose the catalytic site through the cysteine being changed to something else. Real example. I will get back with a real example, so I don't have any right now in my head as of now, but I'll give you a good example later. There's also proteins. Changes in a protein that gets duplicated sometimes changes its location in the cell. For example, this is just an abstract example where if you lose this part of the protein, then you end up being expressed in the heart. And then if you lose the other part here, which is probably a nuclear, this part here will end up being expressed in the muscle. And these are just tags that that allow for certain transcription factors to bind here. For example, the sequence might allow a transcription factor that is important, that is expressed in the heart to bind, but because you to the muscle, but because you don't have it, you end up being expressed in the heart. So this could be an inhibitory region, for example, and then you can see the opposite for the muscle. Because you have to remember that not all proteins are expressed everywhere. We do have about 20,000 genes, but not all of them are expressed in the same way in every cell. So let me just show you an example. Protein expression atlas. So if you are studying a protein, let's say my protein of interest, and you click on here, then you can see, wait, where's the tissue? So if you take a look at this protein here, then you can, wait, let me show you something that's much simpler. Tissue. Okay, so if you're studying this protein, it will tell you where it is mostly expressed. Obviously, don't study it in the eye. Has probably no expression in there. But if you're studying it for, let me do this like this. Right, if you're studying this protein in your cerebral cortex, then probably it does have a function here because you find it as being expressed a lot in this place. If you don't find it in other tissues, probably because it's not there, so I don't know why would you study it in the ovary, right? And so this is important because when you're sequencing DNA, you do find your proteins and all that, but to study their function, it's important to be aware of the tissue in which they are expressed and under which conditions they are expressed. And so you'll need to figure those out before you're able to go on and say their function. Also, their function changes based on the tissue because the set of proteins that are found there will be different. And so your estrogen-related receptor alpha might bind to X protein in your liver, but protein Y in your brain, let's say. And because it does that, its function completely changes. Okay, so this is why functional annotation is very hard. It's not a simple thing to do. Yeah, this is just what I said earlier. Should I put it earlier? Now, computational assays are not perfect because they can miss genes. Because for you to create a computational assay, you need to have you used to have a certain set of rules that you would tell the computer to look for so that you can actually identify these pieces, right? But if your splice sites are different for that gene, so non-canonical means they're not your usual splice sites, then you'll not identify them because you taught the computer to look for a specific pattern, but that pattern is not found there. So you miss the splice site. Introns too large, there's a lot of problems with this. One thing that is kind of new, machine learning, well, not new, machine learning is pretty old, but it's basically statistics. But you can actually train the computer to find patterns in your data instead of telling it the pattern to look for. And so this is why there's a big hype on this AI stuff, which is kind of interesting because now you can just put a lot of data and then tell it, figure out what the hell is happening, and then it will cluster things based on similarities, okay? And then if you train it by giving it a specific, you label it specifically, for example, you say this is an intron, this is not an intron, this is that, then it will kind of make sense of the data by itself, okay? Without telling it an intron needs to have an AG splice donor site and an GT splicing whatever other site, okay? So there's that. There's a lot of new tools that are starting to use this, which is interesting, but obviously all the previous tools that were used don't take advantage of this. So one problem with the tools, they get created during the PhD of a student, and then that PhD student leaves the lab, and suddenly the software is not updated anymore. So when you're using them, sometimes actually most of them are not updated, Okay, usually you finish your PhD. If you really love your project, you'll continue working on it at home. But 99% of the software is there, they get left, and then at some point you go on the website and it says that the website is not working anymore. So you end up with obsolete papers, and then obviously this field is changing so fast. If you see the amount of data that is put online, it's crazy, okay? So, but these are interesting. You can stay at home and build these if you have a little bit of math background. Not even. There are now libraries that allow you to do this quite simply. The expression assays, as I said, they don't always detect the genes in all the experimental setups. And so you need to find the right tissue, the right time, the right temperature, the right whatever to study the function of a gene. Sometimes your gene, you will not find it unless you have, for example, insulin. If the cell has insulin bound to its receptors, then suddenly your gene appears. So then you can study its function. And so if you're trying to study its function, all your experiments will contain insulin, right? But you don't know this. So it's quite complicated to figure out new functions. This is why you use computational to get an idea about your protein. Conserved domains, the issue with pseudogenes. But this is specific problems. Usually most of the genes are the same. Sometimes you'll end up with these differences, and then you will not be sure of what's happening. Right, so what is actually a human gene? I talked a lot about things, but nothing too clear, right? Seems like they have a start site, stop codon, maybe Tata box, things like this. So there's a new tool, well, not new tool, ENCODE, which is the follow-up of the HGP project, which is short for Encyclopedia of DNA Elements. And it looks at the structure of the DNA and links it or associates it to function. There's another group that's also doing the same work. It's called GWAS, Genome-Wide Association Studies. They're trying to also figure out complex functions of proteins in diseases. So this is kind of normal state. And then this is in the disease state. So I'll be talking about ENCODE next lecture. But just to give you an example, like a little bit of progress that they had, in 2007, they actually based their assessment on only 1% of the genome. So that's not a lot. Right after the genome was published from the Human Genome Project. Up to 2020, now they have information from other genomes, for example, the mouse genome, and 2012 is where they had an extensive data set that was added to it. So we'll see a little bit about the ENCODE project, then we'll get into what? Into the selfish DNA, and then the functional RNA, and then it's the 4 interesting topics. So the paper, prostate cancer, the Neanderthal paper, I'll probably talk about some biotech stuff, and then gene therapy, and then it's your presentations.